Potential new strategy to fight ovarian cancer found. A new discovery opens the possibility of developing a novel way to fight ovarian cancer. Researchers at Baylor College of Medicine and the University of Texas MD Anderson Cancer Center have revealed a previously unknown cellular pathway that selectively regulates a mutant protein called p53R175H linked to ovarian cancer growth. The researchers identified a key regulator of this pathway, USP15, and suggest that designing drugs directed at this regulator might lead to better ways to control cancer growth in the future. The study appears in Nature Communications. Ovarian cancer is currently treated with surgery and chemotherapy. Chemotherapy for ovarian cancer is based on drugs that act on any cell that is rapidly dividing, healthy or malignant, which has side effects, said first and corresponding author Dr. Achyut Padmanabhan, instructor of molecular and cell biology at Baylor College of Medicine. If we're able to develop treatments that target only cancer cells, then we might be able to minimize side effects. Research has revealed that mutant p53 proteins might be the target scientists have been looking for. More than 96% of high-grade serous ovarian carcinomas, the most lethal and common form of ovarian cancer, have mutations in the p53 gene. These mutations have been shown to play key roles in the production of tumors and in the development of drug resistance. Normal p53 protein is known for playing a tumor suppressor role. In contrast, the mutations studied by Padmanabhan and his colleagues, p53R175H, can promote tumor growth. Normal p53 is rapidly broken down inside most healthy cells, but p53 mutants, such as p53R175H, are not. They form very stable protein clusters inside cells that promote cancer and induce drug resistance, Padmanabhan said. Researchers have discovered that if we remove mutant p53 forms from cancer cells, the cells will enter a path toward cell death and become more sensitive to chemotherapy. This is very valuable from the clinical point of view. These findings have prompted Padmanabhan and his colleagues and other researchers to find ways to reduce levels of p53R175H inside cancer cells to promote their own destruction. The question has been how to selectively eliminate p53R175H clusters. We do not want to affect the p53 proteins because this also can lead to cancer. Our goal is to find ways to reduce the levels of mutant p53R175 only, Padmanabhan said. We do not know how to do it until we discovered a unique pathway that can regulate the mutant, but not normal p53. Once we identify the specific pathways, we can design drugs to manipulate them. When Padmanabhan began working in the lab of Dr. Joanne Richards at Baylor College of Medicine, they were testing a small molecule called MCB613 for its ability to treat ovarian cancer in a mouse model of the disease. In these experiments, Padmanabhan observed that in cells treated with MCB613, the levels of mutant p53 protein rapidly decreased. This was interesting because it told me the drug was doing something unique to this mutant, but not the other forms of p53, Padmanabhan said. In this work, we tried to understand how treatment with MCB613 resulted in reduced levels of p53R175H in ovarian cancer cells. The researchers found that MCB613 does not decrease the levels of mutant p53R175H protein directly. It reduces the levels of another protein called USP15 that removes a specific tag from the p53 mutant that targets it to undergo degradation inside cells. Having few molecules of USP15 results in more mutant p53R175H protein tag for degradation in the cell, Padmanabhan said. As a result, the balance tips elimination of mutant p53R175H proteins, which results in more cancer cell death. We also discovered that mutant p53R175H is destroyed in the lysosome system of the cell as opposed to normal p53, which is broken down by another protein degradation system within the cell called procedamine.